Hello, welcome to Pharmacology lecture series. Today we are going to discuss antibiotics and their mechanism of action. So what do we mean by the term antibiotics? So these are the agents produced by microorganisms which act specifically on bacteria, and either they can kill the bacteria or inhibit the growth of bacteria. The ones which tend to kill the bacteria are called as bactericidal, whereas the ones which inhibit the growth of the bacteria are called as bacteriostatic. Now, we will look into the classification of antibiotics based on their mechanism of action. But before that, it is essential to understand the normal machinery of the bacterial cell in order to treat bacterial infections. Normal bacterial cell has its own physiology and morphology, which consists of outer capsule made up of cell membrane and peptidoglycan layer. It consists of its own genetic material, that is chromosome, which is unlike human genetic material when it comes to its shape and pattern of coiling. This genetic material is capable of transcription and translation, which synthesizes its own messenger RNA and has the capability of synthesizing its own proteins with the help of ribosomes, that is 30S and 50S ribosomes. So here we can see the proteins being synthesized in the bacteria with the help of 30S and 50S subunits of ribosomes. Now we'll discuss the one more special characteristic of bacterial cell, that is, it has its unique metabolic property which metabolizes tetrahydrofolic acid with the help of precursor para-aminobenzoic acid. So it is very much essential to synthesize folic acid, which maintains the genetic material of the bacteria. And also this feature of bacteria is unique to synthesize folic acid, as unlike human beings, the folic acid cannot be supplemented through diet or any exogenous source. Thus, the bacteria has to synthesize its own folic acid endogenously. So this is the brief introduction to normal bacterial cell. Now, we are going to look into the mechanism of action along with the examples of the drug. To begin with, the first category of antibiotics are the ones which inhibit the synthesis of bacterial cell wall. So now we are going to magnify the cell wall. Bacteria consists of cell membrane, and it also has peptidoglycan layer which acts as cell wall. Now, regardless of bacteria, either it can be gram positive or gram negative, it consists of cell membrane and peptidoglycan layer. However, there can be change in the thickness of the peptidoglycan layer, or there can be multiple cell membrane layers depending upon the type of the bacteria. Now, the drugs which act on cell wall and they will not allow the synthesis of cell wall include beta-lactam antibiotics, which include penicillin, cephalosporins, carbapenems, and monobactams. Apart from beta-lactam antibiotics, one more drug, that is vancomycin. Moving on to the second type of drugs which will disrupt the cell membrane. Once again, we'll magnify the cell membrane. The outer layer of the cell, these drugs attack on the cell membrane instead of cell wall, and these drugs cause pores and gaps in the cell membrane, which leads to cellular leakage. And the examples include polyene antibiotics. And the examples are amphotericin B, nystatin, and hamacin. Second example of drugs that are polypeptides. And the examples are polymyxins, or they are also called as colistin, and bacitracin. So these drugs, polyenes and polypeptides, they target on the cell membrane and they will not allow the synthesis of the cell membrane, and they will also cause the pores and gaps in the cell membrane layer. So this lipid bilayer will be disrupted and the internal components or the organelles of the cell will be leaked out of the cell and the cell will lose its integrity. Moving on to the third category of drug, drugs that inhibit the genetic material of the bacteria, that is synthesis of DNA and RNA. These drugs restrict the certain enzymes required for the synthesis of DNA and RNA. Examples of the drug include fluoroquinolones, nalidixic acid, and rifamycin. Fluoroquinolones include many examples: ofloxacin, norfloxacin, getifloxacin, moxifloxacin are some of the examples of fluoroquinolones, which will act by inhibiting the DNA and RNA synthesis. The next type of drugs are the drugs that inhibit the protein synthesis. We all know that proteins are essential for the maintenance of the life. We'll be using certain drugs which will not allow the synthesis of proteins. To be more precise, we'll be using drugs which act on specifically 30S subunits of ribosomes, which include the examples of tetracycline, gentamicin, and streptomycin. Gentamicin and streptomycin belong to aminoglycosides. And the next category or the subcategory include drugs acting on 50S ribosomes, which include the example of chloramphenicol, clindamycin, erythromycin, and linezolid. Clindamycin and erythromycin are classified under macrolide antibiotics. Moving on to the last category of the drugs, which is fifth category, are the drugs which act by inhibiting the synthesis or metabolism of folic acid. And the examples include sulfonamides, sulfones, trimethoprim, pyrimethamine, and para-aminosalicylic acid. So that's all, folks. So these are the examples of antibiotics and how you can remember the mechanism of action, that is in brief manner. Thank you.